Hello and welcome to episode number 99 for the National Land Realty Podcast, where we discuss all things land. Our goal here is to inform, educate, and entertain those of you who own land or are interested in the buying and selling of land throughout the United States. My name is Mac Christian, and I'm the Chief Marketing Officer here at National Land Realty, and I'll be your host for this episode. Now, I've spoken about this before. It's episode number 99. So that means our next episode will be our live 100th episode, which is cool for us, cool for you, because if you listen to all 100 episodes, you know a lot about land. But we're going to keep going with this thing, but episode number 100 is important for us. Now let's talk about the episode at hand. In our previous episode, we spoke with Jay Cantrell, wildlife biologist for the South Carolina Department of Natural Resources, about chronic wasting disease. Previously, we also spoke with Dr. Mike Chamberlain on the same topic. Now today, it's our special privilege to learn from Doctor Sonja Christensen on the same important topic. Dr. Christensen is a professor in the College of Agricultural and Natural Resources at Michigan State University. She's renowned for her knowledge in deer biology and was recently named to the National Academies of Sciences, Engineering, and Medicine Chronic Wasting Disease Committee. Dr. Christensen is one of the top minds in the United States on the topic today, and she is here to answer our questions. Now, sit back and enjoy. Okay, so I am sitting here with Sonja Christensen, which is, don't confuse it with my last name, because I don't know about you, but I always get called Christensen. But it's a thing, gets bounced back and forth. But you definitely won't get it confused because Sonja is much smarter than I am. So we're going to dive in here. Sonja is an assistant professor for the Department of Fisheries and Wildlife with Michigan State University, and it specializes in animal diseases. Most recently has been named as a researcher for the National Academy of Sciences and Engineering and Medicine to research chronic wasting disease, right? Or for the board, the research is that. And so what I want to let you do is tell your story, because you can do that better than me, and just tell me how you got to where you are and what you're working with now.
Yeah, absolutely. Thanks so much for having me, Mac. I specialize on, I'd say, wildlife ecology, population ecology, and I've in my whole career since I was an intern with Minnesota DNR back in the day, I've worked on cervid populations in particular. So, things like moose or elk or white-tailed deer, Sika deer even. So that's been my specialty. And in the throughout my career, I've been really honing in on disease ecology as well. So my world sort of is a combination of traditional wildlife science and wildlife population ecology with disease ecology and understanding population management relative to what we can do with disease. So, like I said, I'm actually from Minnesota originally, where I got my undergrad at Minnesota State and then started an internship with Minnesota DNR with their research group there. And then I went off to Penn State for my master's degree, worked at the USGS co-op unit there. And that was a great experience. I then it's kind of a tour across the country here, but I then went to Massachusetts, where I hadn't even stepped foot in that state until I became the deer project leader for the state agency there, deer and moose project leader. So in my line of work, there aren't a lot of these positions, and to be a deer biologist, deer supervisor, project leader for that state was a tremendous experience. I had great colleagues at Mass Wildlife. I then decided, after about 3.5 years of working there, I decided to go back and try to tackle that PhD. And get back into academia. I spent a little bit of time at University of Montana working on some elk, and then I ended up coming to Michigan State University for my PhD. And then I've been able to stay on here as an assistant professor. So again, I've worked with cervids the whole time. I think I just captured basically from 2004 until now. So that was 20 years. So this is my jam.
So you just spoke to something that I have never actually even thought about, and I think it's actually kind of poignant to discuss really briefly. Is there's not a lot of roles. I mean, when you talk about somebody who's specializing in deer populations, there's like a couple seats in every state for this, right? Like there's not many.
Yeah.
And you have managed to find a home there. I mean, like you got your place there.
Yeah, I work with a lot of undergraduates and graduate students now. And one of the pieces of advice that I give to wildlife students is if you have any ability to move and get out of your home state and take opportunities that might be elsewhere, try to do it when you can, because it's only harder to move around a lot as we get older and our families emerge and things. So yeah, I was able to chase after some opportunities and yeah, I've been really fortunate that way. So I joke around that I've lived in 5 M states. So yeah, it's been a good role, but it's a small field. And I also tell undergraduates and graduate students that in about 2 phone calls, we can find out a lot about what's going on with past experiences. So, make sure to be professional and, reach out, build your networks, and make sure that you're communicating well because our network is pretty small. But it's a great profession, and a lot of people that have a lot of passion for wildlife conservation and management.
So you're saying that you can't burn bridges in your field?
Well, this probably goes for anybody, right? But it's small, so you'll hear about it.
Yeah, well, and to that effect, like we were just talking before this, and we've talked we've worked with Michael Chamberlain talking about chronic wasting disease, and you know the name and everything. Like everybody knows everybody, and as I've gotten to know other people, I talked to a fish and game gentleman out of South Carolina, and he knew who Michael was as well. And
Yeah,
working with chronic waste, it's unbelievable what a small world it is.
it's very small, very small.
Oh yeah. Well, and so tell me a little bit about, so you were named to the Committee for Chronic Wasting Disease. Can you tell me a little bit about that, what its function is, and sort of what your role is within that?
Yeah, so that's an ongoing panel, basically, and we are tasked with a specific objective, and that's to review transmission and a variety of other pertinent topics, I would say, around chronic wasting disease, sort of at the behest of the National Academies of Science, Engineering, and Medicine. So again, that's an ongoing panel. That is available online in terms of some of the background, who the panel members are. We are still ongoing, so I can't really speak to what's happening among the panel right now. But we're working hard to get a report done and try to work together to produce a product that will hopefully be really helpful and science driven from a National Academies, a very reputable, credible organization around this really challenging disease.
Okay, so I kind of want to dive in, and anybody listening is listening for chronic wasting disease information, so it's best that we get that to them, and I want to take it from like sort of a 101 level of what is it? From a layman's point of view, what exactly are we talking about? Because when I describe it to people, I just say it's a bent protein, and like that's the best I've got. Maybe you can explain it a little better.
No, I mean, a misfolded protein, when you say transmissible spongiform encephalopathy, that's kind of a mouthful. So it is a prion disease, right? And there are other prion diseases that you might be familiar with, or that your listeners might be familiar with, such as BSE, which was mad cow in bovids or cattle, right? And then scrapie in sheep is another one. Humans, there's a human prion disease called Creutzfeldt-Jakob's disease. So, and there's a few other sort of prion diseases out there. Now, and prion diseases are really unique, and that's a lot of the challenge of these diseases, particularly chronic wasting disease, because they aren't alive. It's not like a bacteria. It's not a virus. We can't address it with a vaccine in the same technology and way that we would with a lot of other diseases. In fact, chronic wasting disease does not have any known cure at all. Once a susceptible animal is infected with that misfolded infectious prion or protein, they will ultimately succumb to it and die. So, and that's always the case. And this occurs for a variety of cervid species, so white-tailed deer, mule deer, elk, moose, caribou. And it's scary. It's infectious. We can't target it. There's not a cure. We don't even have a great, approved test for antemortem testing. And when I say antemortem, like if you have a live animal, it's really challenging to get a really good verified, standardized diagnostic test to say that this animal has CWD and this one doesn't. There's a lot of really great work being done on that right now. And so there's a lot of headway in that direction. But again, another challenge. And then, so no treatment,
Well, I was
no
going to
valid
say,
animal.
you
Yeah.
can't even like, and this is to my understanding is you can't even freeze it or heat it up until it disintegrates. Like it's very, very, very resilient.
Yeah, it's extremely resilient. There's folks who incinerate it or incinerate deer samples after they've been looking for the disease. But these incinerators are really, industry, huge incinerators, and typically you
Right.
can't just burn it. And yeah, absolutely, you can't just freeze it. Otherwise, if that was the case, we wouldn't see it in northern climates, right? So it's challenging. And then once it's in the environment, let's say a deer dies or, succumbs to disease, dies in the back 40 somewhere, and long past that carcass, decomposing and being gone, those prions can actually bind to the soil and to be taken up by plants and still be infectious. So long after that, deer that had that disease is gone, you might have a new animal come in and become infected from those infectious prions, the CWD prions, maybe even years later. So it's really challenging because it's almost like an environmental contaminant and this infectious disease that can be spread between deer through saliva, urine, all sorts of different ways. And so it's just challenging from a number of perspectives.
So I want to sort of just dial back and go into the history of it a little bit. So discovered in the 60s in Colorado.
Yep.
Rumored to be like, one of the old rumors is that it was discovered in mule deer that were housed next to sheep that had scrapie. And is there any validity to that, or does it matter at this point, or is there any evidence that it existed before that?
That's a good question. I will first say our ability to detect a new disease sometimes is challenging, right? So you're finding something that's new, and so knowing when it actually first arrived is kind of, you're looking back retrospectively and guessing a little bit. But again, it's fairly speculative. I think that, there's some experts out there. I would recommend like Dr. Mike Miller, who spent his career in Colorado and is really familiar with this disease. He might be able to speak to that more, but really, it's really hard there's no clear smoking gun in this case.
Yeah. Well, I guess the
as far
better
as I can tell.
is it in fact new? Is it, I mean, is it actually a disease, or is it something that has been around and we didn't detect it until the 60s?
It might have been around a little bit before that, sort of quietly, but it certainly is what I would consider an emerging infectious disease.
Okay.
It's clearly started in North America in that area. Now it's made some big jumps, and there's a few ways that CWD can move to a new place. And, we think about sort of a slow spread, maybe some natural movement in deer infecting each other, a dispersing yearling male going to a new area, some migratory animals perhaps moving in around. But that would mean that spread is quite slow, actually. Humans have a role in this as well, right? Because unknowingly, somebody might be, after hunting, they got it on the back of their truck, their carcass, deer probably look fine, or the elk probably looked fine in the field. They bring it home and then, toss out some parts in their, back 40 or whatever. And that in fact was infectious. So sometimes we see new detections in new areas that they could have been there for a while, or they could really be new. Understanding that timeline is challenging, and understanding what caused that is challenging. So sometimes it could be from a human inadvertently bringing it in. Maybe it's from natural movement. There's a captive cervid industry. They buy and sell and trade live animals. So unwittingly, there could be something that was sold into a new facility that wasn't detected as CWD positive. And again, with no really approved live tests, it's hard to find these things while they're still alive on the hoof. So a lot of reasons why we see it as it's spread in the places we have and where we've had some sort of surprises over time. But again, we see some of those like long distance jumps sometimes, and we see, kind of a slow burn through, just being in proximity to a known infection.
Is there any evidence to, and I only bring this one up because I live in Idaho and it's a controversial topic in Idaho, but is there any evidence to predators transferring the disease across state lines? So the one that's the big one in Idaho is wolves, and it's the word on the street, I don't know, rumor, whatever you want to say, but there's a lot of conversation saying that wolves are spreading it throughout the Northwest. Is there any kind of data that yields itself to that?
There are some studies out there from some researchers who are looking at that. And basically, you need to understand in sort of a more controlled setting what that animal ate, if it was knowing that it was infectious in a kind of controlled challenge study, and then sort of what came out the other side of the animal, right?
Right,
So,
right. It's like it's relevant on their digestive tract. If they could hold it and then run 100 miles.
And defecate.
Yeah.
And it's still an infectious protein. So I think that research is really ongoing. I don't think it's conclusive at this time, but there's good, research going on. And then I would also say, humans are eating potentially CWD infected material too. And so what are we doing? So that hasn't really been studied either necessarily. So, thinking about what's moving it around, I think that, there's some areas of research need for sure, trying to understand like cougars or mountain lions or wolves or what those interactions are. There's been some research in crows, for example, other scavengers, flying and then defecating in a new place. So certainly there's some probably other species, wildlife species that may be involved. However, again, I think that we need more science on that. And when I'm thinking about risk prioritization, I'm thinking more about, the big ones that we can control and the ones that are probably the more obvious transmission risks, like moving infected carcasses or live animals on the landscape, or, again, just known infections growing with high deer densities. Those are the things that are more likely to be the case for that direct or indirect transmission.
So one thing that was spoken to me sort of in a hushed whisper, right, is I'm getting all ominous with all my questions here. but so one thing that was said to me was if you see chronic wasting disease hit an area, the first thing you should look at is for like deer or elk farms. And is there a correlation between those kind of setups and where we see it transferred?
Yeah, I think that CWD has a history, right, of, again, a variety of ways in which it pops up. And so there's a really nice website, cwd-info.org from the CWD Alliance. I would recommend checking that out. There's some really nice maps on that website, and there's a lot of really good information from Matt Dunphy and others at CWD Alliance. But when you look at those maps, there's some places certainly that might not have any captive deer facilities, and some other places that, there's a lot of captive deer facilities and that hasn't been detected in the wild yet. But our ability to know sort of which came first when we find a wild detection or a captive cervid detection is challenging, right? And so there's a lot of good reason to make sure that there's a strong boundary between those live cervids that are for the captive cervid industry. They have their own goals and values around why they might have those animals in the farm setup or a high fence operation or something else, and the wild population. As much as we can minimize contact between those populations, the better, and for the sake of both populations on either side of the fence. So double fencing, making sure that those fences don't have a tree that comes down and deer moving between them, good tagging and, within the fence, and identification and testing procedures are really good. So, and then again, state and fish wildlife agencies are, working on weighted targeted surveillance in their free ranging populations to actually try to pick it up on a landscape in a meaningful way if it is there.
Yeah, and I wanted to ask you those, and I kind of put you on the spot with those questions, and I think
it's okay.
those, because because I think when something like this pops up, it's terrifying, right? Like no one's ever experienced it. Everyone's scared, and everyone looks for smoking guns. And oh, it's the wolves. Oh, it's the deer
Yeah.
farms. Oh, it's this, and it's like if I'm hearing you right, and if I'm hearing right the conversations that I've had on chronic wasting disease so far, there's a lot of causes, and it gets spread by all kinds of things, and people are a big part of it. Animals can be a part
Mhm.
of it, and deer are not exactly migrating from Colorado to Mississippi, right? Like that's not happening.
Yeah.
So
Yeah.
something else is doing it, and chances are like a wolf is not doing that. So I haven't heard of many wolves in Mississippi. Crows, I'm
Yeah.
assuming a crow poops before it gets to Mississippi from Colorado, right? So like,
I hope so.
right. So the one thing you have in there is us.
Yeah.
And everybody wants to be the hero of their own story, and nobody wants to say it's people. So
Yep.
there's a lot to the equation, right? Like when we're looking at this,
Yep.
this is a pretty widespread thing. And I mean, how many states are we in now for chronic wasting disease?
Gosh, we're in the 30s.
Is it in the 30s? Okay.
Yeah, California just popped positive very
Oh,
recently.
I did not know
Yep.
that.
Yep. I think let's see here. When was this? Just within a month. It was, or so. It's been very recent.
Oh no.
So, yeah, I'm trying to find a date on the press release. Maybe May 8th.
Oh, very
Or
recent.
something was,
Okay.
yeah, early May. So, and the positives were found in places where it somehow skipped all of Nevada. So, that's not really pointing to, a natural spread from a known
That
outbreak, right?
that sounds like
So,
people, right? Like,
yeah, yeah. So some of the research on chronic wasting disease that I do. So again, I focus on sort of deer diseases, and a lot of my traditional training has been in population ecology, using statistics and spatial modeling, a lot of behind the computer, and also a lot of field work, radio collaring deer and other species to understand movement and what they're doing, who's risky in the landscape and such. Collecting ideas about like deer abundance, even just so we know how many are out there and how is this disease impact of a variety of diseases impacting a population. But yeah, one of the more recent areas of research I've gotten into is actually doing more what we call human dimension surveys. So shifting and building some of my research repertoire around humans and asking them questions. Because we can learn a lot about what's going on and attitudes around or challenges to sort of uptake and risk mitigation behaviors that hunters or landowners might take, and how we can facilitate that a little better. One research project I had, actually in collaboration with National Deer Association and a few other researchers, was looking at hunter behavior specifically and how we can work on messaging to get hunters to maybe take up some practices that will hopefully reduce the spread of CWD in their areas.
got you. And I'm curious about that too. So when I want to talk about sort of the biology really quick of it, and then I want to dive into that.
Yeah.
But the you talk about the live testing, that there's not really a good way to live test, and I'm assuming that that's because you usually need to do the testing out of cerebrospinal fluid. Is that
Well, there's actually been a variety of technology, and you might be a little horrified for what I am about to tell you of how
okay?
we get some samples. But there are tonsillar biopsies. That's one way that, as deer biologists, we, with a live deer in hand, often we're putting a radio collar on it or some other thing. But you can do a tonsillar biopsy to look for CWD. You can also do basically a rectal biopsy. There's lymph nodes in that rectal tissue that would contain the CWD infectious prions. And then some folks there's a few different methods out there. Those are kind of some of the more common ones. There is an ear punch that's so basically ear tissue, which is more, tissue and muscle and such, that's being used. And there is just really, I can't emphasize enough, I probably can't even mention all of the studies that are sort of in progress right now that I couldn't really even speak to directly that are actively working on this, because this is such a linchpin for being able to better manage this disease. So there's a lot of work on trying to improve that diagnostic test and get something that's.
Do we know? Do we have anything conclusive on how it replicates?
Yes, there's a lot understood about the prion, the CWD prion. I think that it's impressive how basically one infectious prion can contact or talk to, in a way, another protein in that is not infectious or not misfolded, and basically create that next one to become infectious as well and misfolded and sort of in this infectious form, this CWD prion form. So a lot of the microbiology that is at play there is probably best left to a prion biologist and not me. But yeah, this is happening, and it really is targeted in the central nervous system for the most part, in the lymph nodes and such. So those are areas of in terms of the anatomy of the deer, the elk, that we really are going to focus on as being really infectious material. So, venison or the meat itself is probably less risky than where, like you said, the nervous system or brainstem or lymph nodes or other tissues associated with that system.
Yeah, I mean, it seems like it seems like it's hijacking the cell and like getting it to replicate like, is it like inserting itself into like a ribosome thing? I don't know. Like
it's again, it's a protein, right? So it just needs to touch another protein.
So it links up to other proteins.
Yes. Again, I think that I'm going to get outside of my lane real quick.
Yeah. No, I get you.
But yeah, I for wanting to be very accurate, I would say the prion biology is pretty well established. But yeah, it's very infectious, and it doesn't. It's protein, right? So it doesn't need to deal with DNA. It doesn't need to. It just needs another protein to infect. So.
Oh, okay, I got you. That's interesting. I mentioned wanting to circle back to hunter behavior and what hunters can do. So tell me a little bit about this on what sort of has been what you found and sort of what you were trying to teach to hunters.
Yeah, so we did a research project through an online survey, and what we were looking at is we produced about 5 2-minute videos. Kind of think of them as like public service announcements for hunters on some things that would be helpful to help us manage CWD in free-ranging populations. So things like getting your deer that you hunted tested, right, and turning that sample in, properly disposing of carcasses in landfills appropriately and such, wearing PPE, targeting, which is not always very popular, but targeting female, like does or female elk, or yearling males even, which is not always very popular. In addition to that big buck, because we want to sort of keep deer densities at a reasonable level, so we aren't having more infection with really high transmission rates with really high deer densities. A few other things we're actually talking, we call it sort of myth busting around the deer camp. So talking to your buddies at deer camp about what CWD is, and if you hear somebody saying something that it doesn't line up with the abundance of science and information that's out there, say, oh, hey, actually, I heard this, and sort of being that voice. Another one was asking processors about whether or not they mix venison in one batch, or if they just do your deer one at a time, just to reduce risk to yourself, basically, so that you're not worrying about all sorts of deer that might have been CWD positive running through the same processor. So we did that by sort of demonstrating with videos and these 2-minute videos, like the intended action. So if you sort of see something and can visualize it, or, we actually had some folks kind of act out a skit of, talking to a processor. So, to give folks some words to maybe use if they weren't sure how to start that conversation. And then hopefully, enact that in their practices when they're hunting out in the field. Now, we did a survey, a sort of a pre-intention, like, what do you think about this? What do you typically do? And then we showed them the video, and then we followed up in the same survey with, all right, now what do you think about this? What do you intend to do? And then one piece that we did that not a lot of researchers are usually able to do, we then followed our sample of folks who did our survey to the end of the deer season. And then we surveyed them again and said, what did you actually do in the field? And of course, for those folks, it had to be people who were successful in harvesting an animal. It wasn't really applicable for those who didn't. So that reduced our sample size right there. But we still had about 1,400 responses from people who filled out all of those points of our survey to the end point. And we found that our videos, our information treatments, did improve the intention of people. They were educated and said, oh yeah, I will do that when I'm out in the field next. But unfortunately, and these were all done in, early October, so the beginning of most deer seasons. But by that February, when we then followed up with our endpoint survey, most people had actually didn't change their behavior. They'd actually kind of reverted to whatever they had stated they had done the previous year. So it's really hard to change those ingrained behaviors when we're out in the field, what we've been taught, what our, custom is when we're out there, hunting and field dressing an animal or something. So I think that there's some ways to keep chipping at that. But yeah, it's tough.
Is that link open to, is it available?
Actually, I've reported out on this for our funding agencies, but the paper is in draft form, and I hope to get that published here very soon. So
cool. Well, if you end up with the video link, I would love to put it.
Absolutely.
What are some of the myths that you address in the video that sort of are your hot button topics that you kind of deal with and that you want to get off the table?
Yeah. Well, I think, gosh,
there's probably a lot.
To start. Are there any that you've heard? So I think knowing that it's a prion first is always important. Knowing that it does affect deer. Just establishing CWD will kill the deer that has it. That is case in point. And that wild deer aren't going to just recover. So far, when we've seen deer populations that have very high prevalence of CWD, it doesn't look good. And you probably heard that from Chamberlain and the Arkansas study. But they've got really high prevalence, and there's a lot of mortality happening from CWD directly. And that's after they ruled out scavenging and mortality and all the other things that deer die from. So that's one thing. Folks just thinking, this isn't real. It doesn't really affect deer. It's not really going to affect the deer population that I hunt in or that I see on my property. And maybe they're non-hunters. They just like to see deer. But this can really have a damaging effect on deer populations.
Yeah, and I think what a lot of people dial into is the inconsistency of it. And I think Michael talked to that when I spoke to him, was that you see some populations where the prevalence is like 20% up to 50% that they'll see it, and then in some populations you've got like 2%, and some of them seem to be resistant naturally. So he said that there's some theories that it could be a biological thing, like their genes could be just have a natural resistance, or they just don't know.
Yeah, there's some research out there. A lot more research needs to be done in this area, and I would say it's only sort of partially relevant for wild populations and how they breed and how genes are mixed in wild free-ranging deer populations anyway. But there are some alleles that seem to be more resistant to getting that infection in terms of how quickly they get infected. However, even if it's slower, they still ultimately die from it. So in some cases, you can have some animals that might have some a slower time until they are really clinical or have a lot of what we would call sign of the disease. But they're still shedding prions actively that whole time, even though they look healthy. So it's still tricky.
So you just said
it's a challenge.
You said alleles, and I want to make sure we clarify what that is. That's like a genetic kind of genetic strain or inheritable
genotype. For something specific. So we all have like genes for hair color, but your particular allele might be red hair.
Yeah. So when we're talking about it, so when you say that there are some alleles that seem to be resistant, what we're saying is there's some, like if you inherited something from dad or something from mom, that might help you be more resistant. Kind of like, I guess, kind of like what we see with, wasn't there was a correlation that they found between bubonic plague inherited families whose descendants went through the bubonic plague were more resistant to like HIV. Was that the?
I haven't heard that, but that could be the case.
Something along those lines. But I mean, it's same concept, right? Like it's something you inherited making you resistant.
There's a lot in our genomes that can be related to our ability to be susceptible to a variety of different types of diseases. So certainly that could be the case. But there's a variety of things going on there. Back to the myths. Were there any other myths that you kind of needed busting?
Let's see. I'm trying to think off the top of my head. If it tests negative, it doesn't have it.
That's a good one. We just say it wasn't detected to have it. So tests aren't perfect. And there are some that throw out false negatives and sometimes even false positives. But that's certainly one too. So I think, just in general, knowing that it kills animals, and I guess getting back to your variation and why we see 2% in some places and 20% in others. I tend to think that where we see those really high prevalence levels, even though they being the state agency or an area detected it relatively recently, that indicates to me that it's probably been on the landscape there for quite some time. We see basically prevalence curves go up pretty quickly. It's sort of this disease sort of in the environment stays pretty low for a while often, and you want to keep it there. 2% is great. If you can keep it at 2%, and that's a lot of what state agencies, fish and wildlife agencies are trying to do, just keep it low where they've got it. Contain the spread where they've got it, because you don't see too many really terrible effects when it's really low like that. But then when it starts shooting up and really that transmission rate really increases within a herd or a population, then you can start to see these really high prevalence rates. And that's where you start to see more mortality directly associated with the disease. So it's usually just somewhere along that prevalence curve, unfortunately.
So I was going to say, what we're probably looking at is date of introduction as far as areas where it's if you let it get away from you, it's probably going to hit that higher density infection rate than, say in an area like, I'll just use Idaho for an example because I know it. And where a lot of the area is looking at like a 2% infection rate, but it just hit here. And if I understand it right, it can take a few years to actually test positive. Once it hits a population and they have it, it's got to build that protein load to be detectable, right?
Yeah, it was some of the more sensitive assays out there. Like there's one called RT-QuIC, which is an acronym. But again, there's assays out there, diagnostic tests that you can detect a pretty low level of infectious proteins now, which is good. What often likely happens if a state agency is doing surveillance and they find it and the prevalence is really low and they're looking hard for it, so they have a pretty good understanding that they're sampling adequately, which is hard to do, quite honestly. And then you got it early, which is, again, terrific. That's the best case scenario if you do find it. And then trying to contain it and keep it low and keep those densities low. There's places with habitat differences. Western states, habitat's different. white-tailed deer versus where mule deer are versus where elk are. All of those are going to have a little bit of different risk factors in terms of the ecology of those host species. And when I say host, I mean like the deer, or the elk itself. So that ecology really plays into that risk and where we see prevalence that might be higher. East of the Mississippi, you're mostly focused on white-tailed deer there. There's definitely some elk too, and moose, but it's a little bit different dynamics and habitat structures there that might allow or disallow in some places transmission among animals.
What, from a data standpoint, or if you've seen anything from a data standpoint, seems to be the most effective mitigation strategies? Because every state's kind of on its own, right? Like they all kind of experiment, they all do their own thing. And what was it, a few years ago? I think a group in Pennsylvania was trying to say it was a bacterial infection. And
oh yeah, that's definitely a myth.
Okay, you're the first person that knew exactly what I was talking about with that one.
Yeah.
Everybody else I've asked with that. So tell me a little bit about that and what they found.
Well, there's a particular researcher who is really focused on that particular theory. And I think that as a scientist, I always encourage more science. Ideally, good science is replicated, right? And you're testing something and trying to falsify it, right? And so there is that researcher who is pretty focused on it being a bacteria that causes CWD. However, the preponderance of science and data so far would strongly suggest that this is a prion, in fact. So
I appreciate your tact there.
Well, again, I try to focus on just good science and focusing on that. Yep.
And to that case, there is always the possibility that something is found or whatever, but it's like, it has to be, if you're going to throw something out like that, you better be able to reproduce it. And
yes,
so there was that. And I have been told, this is secondhand. I'm really good at this whole secondhand information that I haven't actually dug into. That's why I like to put people like you on the spot, because these things. But so I have been told that, like in New York, was it New York that had one of the most effective mitigation strategies, and basically they just went scorched earth with the deer populations, but that did stop it, to my understanding.
Yep. So, okay, to speak to mitigation strategies, again, free-ranging wild populations of cervids, right? Because behind a fence, it's more straightforward, but and challenging still, but free-ranging populations, there have been some successes with very surgical targeted removal of animals when state agencies, Fish and Wildlife agencies, have gotten very lucky, quite honestly, in finding it really early. And they had good surveillance, and they were able to figure out, okay, here's 1 or 2 positives on the landscape, hammer it hard, get those sick animals off the landscape. That unfortunately is going to mean culling other animals and really reducing the deer density in an area heavily to reduce transmission. Again, you think of like nose-to-nose contact. They can't wear face masks and they can't social distance, right? So you want to reduce that deer density very quickly. And you then are just hoping that you really caught it early and that it was really at a very low apparent prevalence level. And then there's a few places that have gotten lucky. So that's when you get in there early and you can be aggressive, it's tough. It's not a easy pill to swallow because we want to see our deer. We don't want to remove our deer that seem healthy on the landscape, but sort of for the good of the herd, it's really the most effective thing to do.
So along those lines too, if they're eradicated in certain areas, so like I'll bring up the New York case, right, where they they basically put everything down in that area and it halted sort of the progress. Is there also still risk that resurfaces because it can go into the ground and it's in there in the fecal matter, urine, saliva?
Yep, that's certainly a risk. So, Kristen Schuler is in actually New York State. She's a a professor at Cornell University and a terrific researcher on this, and her in partnership with New York DEC, I think they've really done a good job with very risk-based surveillance. So think of surveillance for a disease. It's, you don't have unlimited resources. So you've got maybe 100 samples that you need to take as a state fish and wildlife agency. You don't have a lot of funding for these things. You got to put them in the best places possible. So with some of these risk-based or weighted surveillance approaches, you can really target where you're looking. So they're really targeting the place where they knew it was. Again, they were a little extra fortunate because they were able to tie a lot of the positives to one focal area, and I believe a taxidermist or a rehabilitator, or maybe I think it was both, all in one facility, area. There were also free-ranging deer, of course. So it was very local, is what I would just want to emphasize. And again, they got pretty lucky probably that they got in there early. They also had some ongoing research from deer researchers at SUNY Syracuse, I believe, at the time, who had radio-collared animals out. So they had a good understanding of what white-tailed deer were using on the landscape, what were the preferred habitats, what was sort of the resource selection looking like there, so that they could focus in on areas where they would be most successful in terms of deer ecology.
I did just think of a myth that I did want to throw at you because I think it's fun. Are you a part of a collaborative that is getting together to try to eradicate the possibility of people harvesting their own food and make them more reliant on purchasing food?
I have the opposite goal probably in mind. So no, I'm not. I would be impressed if there was something that was that organized and who is paying them. But no, unfortunately, us fish and wildlife biologists are kind of the lowly, scientists who are doing it out of passion for animals and healthy populations that ideally can be harvested by sportsmen and women for the probably best free-ranging protein source you can get. So I'm very, very much on the other side of that equation.
Any evidence to it being man-made and spread by people for the same purpose?
Nope.
Okay.
Again,
they popped into my head. You asked me if I had any other myths, and I had to throw them out there.
Hey, you might as well. Again, I think that venison eating is not, putting out businesses as far as I know. So, yeah.
I was going to say, in that scenario, the bad guy would be the ranchers, and that seems contradictory to everything that's talking about.
Now, and hunters, honestly, fish and wildlife agencies, wildlife professionals, we rely on hunter license sales are really funding a lot of the really good research for wildlife populations that's happening, everywhere. So those dollars are matched, and they support turkeys and bears and rabbits and everything else. So those deer license sales are actually incredibly important. So as part of the funding model of fish and wildlife professional work.
So you're saying if you were spreading false information about chronic wasting disease, you would essentially be putting yourself out of a career?
Yes, that would be true. As a deer biologist, especially, yes.
I love bringing into politics into this, because it's science has gotten very political in the last decade, which I know is I'm sorry. Yeah. I'm so sorry.
Well, I think that what I focus on is doing good research that answers questions that are important, and those that are important for having healthy, sustainable fish and wildlife populations. So I try to keep it pretty simple, quite honestly, and just do good work and good science. So
I can appreciate that. So bigger questions, kind of wide ranging. How fast is this spreading nationwide? Is it like, are we in panic time? Like, are we looking, you always see the articles talking about the zombie apocalypse for deer. Like, is that kind of what we're looking at? Or is it somewhat predictable? Or, you're looking at things from like the overhead view, especially from population densities. What are you seeing?
Yeah, not apocalypse. They're sick deer, but not zombies. But this disease is interesting because I study a variety of other wildlife diseases too, things like epizootic hemorrhagic disease or blue tongue in deer. which can actually kill quite a lot of animals very quickly, but then it
I've
sort of
seen
comes
that
and goes.
firsthand. It's nasty.
Yeah, that's one I do a lot of work on. But, as a lot of diseases go, one of the interesting things with chronic wasting disease is actually quite slow. It's a slow progression in the animal, in the individual animal, and that makes it a challenge because, again, an animal could be infected with chronic wasting disease, and you would not be able to tell for months at all. In fact, healthy bucks getting brought in check station stuff, pop positive for CWD regularly. So we would never suspect that, which again is harder if you have a sick and dying animal that's infected and becomes sick very quickly and they fall over, they can't transmit it much further, right? So it actually helps. It's a different dynamic in terms of how we're able to identify disease and how transmission can happen. So that transmission can be kind of slow. It's a slow progression. Again, there's different ways that it's sort of brought into new areas. Some might be some natural spread from wildlife. Some might be from humans. It's really hard to know exactly which one it is. So over time, it's been relatively slow. With that said, if you look at some of the maps in the last 10, 15 years, as people look for it more, they're finding it. And so some of these maps look like they're blowing up. And there's some terrific resources, again, on the CWD-info.org site, CWD Alliance, and also USGS. Brian Richards, who is at USGS and does a lot of the CWD mapping there, has kept up a lot of these maps. As soon as a county pops positive or is detected to have CWD, the whole county gets shaded as in a gray color or something, so it looks big fast. But within a given county, it might just be in one little corner. So again, there's a reason to think that there's still some hope in trying to snuff it out in a few places if possible, or at least contain and control that spread wherever possible. So again, it's generally speaking, as diseases go, it's fairly slow moving, but we're seeing it spread for sure in a lot of places.
Yeah, I spoke to being in an area in Idaho where it just hit, and it's the one valley I've been hunting for like 10 years. They had just 6 years prior to chronic wasting disease had blue tongue as well. And
oh yeah,
that was there were bodies laying in streams, and I mean it was not a sight that you wanted to see.
Yep.
So the last question I'll hit with is the big one, is transfer from deer to humans. What is being looked at and discussed as far as there's no recorded cases? And I actually contacted you because and I think it was just bad reporting, but the Creutzfeldt-Jakob's disease outbreak in Wyoming spurred a lot of people because it was hunters. I don't think they've established whether it was causation or correlation kind of thing. I think they've established it was bad reporting, but along with that, that's it. I think everybody's waiting with bated breath for that headline to hit. And I wondered if there's any kind of consensus thoughts. I know it's probably being discussed. There's the monkey study where it was injected directly into the spinal cord, but that doesn't really factor into digestion or anything. What sort of what are you seeing? What's the discussion in the scientific community?
Yeah, so there are a few folks who really focus on this area of zoonotic potential and what we would call the species barrier, like how quickly or easily can this move between species? And there's a lot of different species that are looked at, but generally speaking, there is not any clear evidence that this has ever been seen in humans. That recent report of the two people who were potentially had Creutzfeldt-Jakob's disease, it was speculation that that was a connection. So I wouldn't even say correlation, definitely not causation. There was just a lack of information there, or a lack of data altogether to put those things really together. With that said, there's a few different studies specifically using macaques as a species model, and generally speaking, the published literature has indicated that there's a fairly strong species barrier to humans or human-like animals at this time. There is some ongoing information of a macaque study that where some macaques actually ate infected venison. But again, that study has not been published, and I think more research again needs to be done in this area, and we need to keep looking. There's some really great work out of University of Minnesota, their organization CIDRAP, and folks there. There's great work out of Case Western University. There's a lot of colleagues looking at this, but again, it's not clear that this is the case yet and that it's really imminent. With that said, I always get my own deer tested and do not eat CWD positive animals. So it's good to be careful and mitigate our risk whenever possible, because there could be a new strain, there could be something different in the environment that shifts a little bit, and we don't want to test that too much.
I've been having more and more conversations because I've been interviewing more and more, people that know the chronic wasting disease. And so I'll get asked, what should I do? And it's like, to me, no matter what, you get it tested, because 100%, usually the first person that gets it gets the disease name, and I'm determined that there not be a Matt Christian disease out there. Like,
I hope that's the case.
Right? Nobody wants to be that first one. And so it's, for me, it's like, okay, and I guess that's a good question to throw back to you. And I know we're closing in on time, and I want to be conscientious of your time. How important is testing in the overall spread or mitigation strategy?
It's very important. I would say a goal for surveillance that a state agency has might look one way, and they might not need samples from everywhere where they're getting them from, because hunters might be bringing them in as sort of a informal, not authorized, food safety test. So I would certainly, as a hunter, want to have my deer sampled and tested for CWD for my own peace of mind, but that isn't always the need of the state agency. So there's different avenues where the state agency needs samples from in maybe a new area, or depending on their surveillance goals. Hunters could really be contributing to helping them understand CWD in those places with that, again, sort of targeted surveillance or weighted surveillance based on risk. In other places, if the local DNR or Fish and Wildlife Agency doesn't particularly need samples, but somebody just wants to get them tested, there's more options for that as well. I would strongly suggest contacting whatever your state Fish and Game or state, DNR wildlife agency is, to see what those options are in your jurisdiction. Tribes a lot of times have options for this as well, different jurisdictions. So depending on where somebody is hunting, I would look into what the options or the recommendations are in those places.
Awesome. Anything that we haven't covered that you think we should?
Not enough time to talk about probably now. It's a big topic. I could talk about deer and deer disease for a long time, but I think these were really good points, and I hope it was informative for the folks listening to the podcast and people who are concerned about chronic wasting disease.
Awesome. Well, I just want to very heartfelt give you thanks. Thank you very much for taking the time.
Yeah, thanks for having me on the show.
Yeah, no, this is great. I really appreciate it, and I want to let you get back up to your day.
Terrific. Thanks so much, Mac.
Bye. This concludes episode number 99 for the National Land Realty Podcast, discussing chronic wasting disease with Doctor Sonja Christensen of Michigan State University. You can learn more about land ownership and the buying and selling of land at nationalland.com. And do not forget to tune in live for our 100th episode. We'll be announcing that shortly. Thank you, and have a great one.